In this tutorial, we're going to explain how to present the results after performing dissection. In the previous video, we've dissected a pig's heart. What can we do afterwards? Well, this practice provides us with an opportunity to present the results in a format other than tables and graphs. Actually, you can take a photo of the dissected heart and stick it to your lab manual. Then you can label it just like that. In addition, you can develop your observation skills by accurately drawing the structures that you can see. As you know, previously we learned how to draw cells observed under an optical microscope. And if you remember, we followed a list of rules to do so. Also, in this case, you should follow several rules while drawing a heart. Some of these rules are: 1. Use a sharp pencil. 2. Don't shade or use color. 3. Use unbroken lines. And 4. Label the structures and include brief annotations. So what else can we do? Well, you can write a description of structures that you observed and relate them to your biological knowledge. For instance, the ventricles of the heart have thicker muscular walls than the atria, since blood is pumped out of the heart at a high pressure compared to the atria. However, the left ventricle has a thicker muscular wall than the right ventricle due to the higher forces needed to pump blood through the systemic circuit compared to the pulmonary circuit.